Hello, everyone. Welcome to the 2024 3-Minute Thesis Finals at Yale. I'm Lynn Cooley. I'm the Dean of the Graduate School, and this is always an awesome event. So I'm very glad you're here, both in the room and online. So I want to start off by thanking our Assistant Dean for Professional Development, Suzanne Young, for organizing this. And I think you had help from Jacob Gonzalez, is that right? Yes, wherever you are there, Jacob, thank you. So the 3-minute thesis epitomizes, we hope, the essence of graduate education, which is, in my view, mastering the ability to convey complex ideas succinctly. We all have to explain what we do to a lot of different people who may or may not have expertise in our fields, and being able to transmit the excitement of what you do and the importance of what you do and the impact of what you do is just incredibly important. And this is true whether you stay in academia or you venture out into some other path after you graduate. So today we're here to celebrate some truly exceptional students and they're from the graduate school, and they will be showcasing some wonderful research from the humanities, social sciences, biological sciences, physical sciences, and engineering. In other words, a broad swath of the graduate school. I want to say congratulations to all of you for making it to the finals and for all the hard work you did to get to here today. So let's give them all some applause. Okay, so what do we do next? We have a judging panel that I'm going to introduce you to, which includes myself and 3 esteemed alumni of the graduate school who bring extensive experience and expertise from a range of fields. So each of our judges has a remarkable story of applying their graduate education to their careers. I'm going to give them a chance to tell you a little bit about that. So what I'm going to do now is just briefly introduce our judges and ask each of them to tell us a little bit more about themselves by telling you about their path since graduating from Yale, a few words, and importantly for today, comments on the importance of communication in their career paths. So I'll let them think about those questions for a minute, putting them on the spot, right? And introduce them to you. So first, Alicia Ellis. Alicia received her AB in German Literature and Women's and Gender Studies from Amherst College, and then her PhD in Germanic Languages and Literatures from Yale in 2009. She's currently Associate Professor of German at Colby College, so she drove down here from Maine. Next, Ethan Lerner is here today. Ethan earned his bachelor's degree in chemistry from University of Pennsylvania, followed by his MD and PhD in immunology from Yale in 1982. He's now the director of the program in itch at Massachusetts. So if you have Massachusetts General Hospital, sorry. So if you have an itching problem, he's your guy. And Associate Professor of Dermatology at Harvard Medical School. And finally, Laura Stevens. Laura earned her PhD in experimental pathology from Yale in 2017. She was a postdoctoral research fellow at Dana-Farber Cancer Institute, where she studied stromal-induced heterogeneity of tumors and epigenetic regulation in therapeutic resistance. Sounds great, huh? She now serves as a senior scientist at AstraZeneca. So, Alicia, Ethan, Laura, I invite each of you to come on up here and just tell us a little more about your career path and the importance of public communication. Thanks. We'll start with Alicia, okay? And I'll just pass this to you.
Thank you for welcoming me here today. As Lynn said, and I'll repeat this again, I think it's important to be able to communicate and distill your projects into the most succinct and salient and coherent narrative possible so that you can speak to any expert or layperson. And in that same line, I would say also develop advanced skills that go beyond your academic research, and be able to evaluate that kind of information and knowledge, and learn how to convey your findings effectively to different types of professional audiences. And I would say that clarity is more important than cleverness, and be able to be really versatile and nimble in your career. After I left Yale, I actually left Yale in 2006 for a dissertation fellowship at my undergrad institution, and I changed my dissertation topic and I changed my advisor. I don't recommend that. And it took me an additional 3 years to finish because I was working full-time at Hampshire College for 8 years. And then I left Hampshire in 2016 and went to Colby in Maine. And it's been a really exciting journey. I love what I do. I love working with undergraduate students. And I would say that in terms of public communication, how that has helped me is that I didn't know that there were skills that I had until I was asked to speak about certain things. And so there were opportunities I never pursued, or I would never have pursued unless I was really asked out of the blue to do something. And so things that were kind of not in my wheelhouse. And so I think you have to be willing to take risks and really put yourself out there and do things that are scary. Thank you.
Ethan.
And that was much more elegant than anything I can say. But so I never grew up, and sort of stagnated at about age 8. And therefore, in terms of communication, I've always thought that it's important to be able to communicate things at about a 3rd grade level to a fairly broad audience. And I mean that, to keep things fairly simple. I had eczema or atopic dermatitis as a kid and would play with bugs, so I was always interested in itch. And then when I was in immunology graduate school here, we didn't know how to work on that, but after leaving, I still wanted to pursue that and realized that immunology was just too complicated, so I became more interested in the neural aspect of things, because neurobiology is very simple. Right. But I want to make a comment about mentoring, which is so important to have mentors, and I've had several. My first major mentor was Charlie Janeway, who was my MD, PhD, or graduate school advisor here. And Charlie's no longer with us, unfortunately. But one of the things that we would do together, or sort of separately but together, is we would look under the microscope individually, and we would serve as each other's control. There was that much trust between us, and that was very important, to just not believe your own data, but to have someone else check it. So that's one thing. Another mentor is a woman by the name of Joan Stites in MB&B here, and we used to go from the medical school swimming over. She would drive, and I would either get a ride with her or walk over to Payne Whitney Gymnasium, and we'd go swimming. And then another is a fellow by the name of John Parrish, who went to medical school here and was head of dermatology at Mass General. And one of the things he taught me and us was, when you're relatively younger, you're kind of on the take. It should be a bit about you. You need to produce. But as you mature, grow up a bit, hopefully have some degrees of success, it's no longer about you. It's about helping others. And you can help others, of course, along the whole way. And then last is a fellow by the name of Steve Katz, who was head of one of the institutes at the NIH. And I was working away on a project, spending all sorts of hours. And when my first kid was born, this is in 1990, Steve Katz says to me, we happened to be talking on the phone for some reason. He said, "So, Ethan, now you actually know what's important in life." So it's not just your work, it's family, it's people, and it's communication. And so keep all those things in mind. Thank you.
Hi, everyone. I'm Laura Stevens. I'm a senior scientist at AstraZeneca, and I'm still what I would consider an early career scientist. And I think being able to communicate effectively has helped me at every stage within my career. And being able to show how passionate you are about what you're presenting and the soft skills that you've learned within your PhD can leverage a career opportunity or help you network and get you to the next spot in your career. So I just transitioned from my postdoc about a year ago, and while the data I produced was important to get me my job, it's really about those soft skills and how you present yourself and whether or not that you can convey your enthusiasm for your topic. I think those are what's really important. Yeah, so I'm really excited to hear your presentations today. Yeah, that's it from me.
Okay, I know what you really want to hear is our finalists, not us. So what I'm going to do now is hand things over to our hosts for the competition, Joe Macheski and Roxanne Morris. Joe is a sixth-year PhD candidate in environmental engineering, and Roxanne is a sixth-year PhD candidate in microbiology. I've worked with both of them for a long time, and I'm so glad you're here to do this. So thank you, both Joe and Roxanne.
Thank you so much, Lynn, for the wonderful introduction, and thank you to our judges for being here today and introducing yourselves. I'm going to briefly go over the schedule for today's event, and then Roxanne will give a brief introduction about kind of how each presentation will go. So first, our 8 finalists will present for the first hour. For our in-person audience, please remember to silence your cell phones now. We don't want to distract them when they're giving their presentations. After the presentations are over, I will invite everybody in the audience to vote for their favorite presentations for the People's Choice Awards. While we vote, the judges will leave the room to choose the first, second, and third place winners, and we will enjoy a performance by the Citations, a graduate student a cappella group. To conclude our event, Dean Cooley will announce our year's winners.
So as you all know, this is a hybrid event that is being recorded, and so just take that into account. We ask you not to share the voting instructions with individuals who are not present. We want people who are either in the Zoom room or with us in person to participate in the voting. So as our presentations start, I will briefly talk about the event guidelines. So each participant will be called up when it's their turn, and we will introduce you and put your slide up on the screen, and then the participants will have 3 minutes. Ming, who is here, who is our other GSAS Professional Development Fellow, will be indicating 30 seconds with the yellow side of the panel, and when 3 minutes are finished, the presenter should stop, and that will be also taken into account during the judging period. Then we will leave the slide up, and the judges will have 90 seconds to take notes on the presentation so that each participant has an equal opportunity. And now I'd like to introduce our first presenter, Theodoros, who is a third year in the electrical engineering program. And Theodoros will present fortifying quantum clouds and securing information in the quantum era.
Quantum computing has become increasingly important across various industries like finance, pharmaceuticals, automotive, and defense. This technology is so crucial that the US government alone has announced a massive $34 billion investment in it by 2023. This highlights the urgent need to protect people's ideas and secure their inventions in this new era of computing. My research focuses on developing novel ways on how to secure the intellectual property of the quantum computer users and to keep their data safe. Right now, if you want to use a quantum computer, you have to go through a cloud provider who plays the middleman role between the client and the quantum computer. As a client, you submit your data to these cloud providers, and they execute them on the actual quantum computers. However, the concern arises when these quantum algorithms, which contain your sensitive information, are translated into pulses to operate the quantum computer. To explain a bit more, When we use quantum computers, we write down some code instructions that all together form a quantum algorithm. These instructions are translated into signals, which are called pulses, and they simply tell the quantum computer what to do. If a malicious provider intercepts and analyzes these pulses, they could potentially extract sensitive information from the quantum computations. So what's the solution then? Well, on the right side of the screen, you can see my proposal. I have developed a new way to hide these pulses using an obfuscation algorithm. Think of the obfuscation algorithm like the PIN pad when you make an in-person purchase with your credit card. Traditionally, the fixed layout of the digits on the PIN pad made it possible for someone to infer your PIN just by observing the positioning and the movements of your fingers. But newer PIN pads have randomized the layout of the digits. This makes it much harder for someone to spy on your PIN, as they can't simply deduce it from your finger movements. Similarly, in quantum computing, our obfuscation algorithm scrambles the original pulses by adding decoy ones in a way that makes it extremely difficult for anyone to decipher the instructions and extract sensitive information, even if they are trying to spy on the pulses. The goal here is to provide safe quantum computations in this new and emerging quantum era. Because the security of information stored in quantum computers directly impacts critical applications across various fields, our commitment to fortifying quantum clouds becomes not just a matter of data protection, but a cornerstone of progress and innovation in every sector that relies on the transformative power of quantum computing. Thank you.
Thank you. Thank you, Theodoros. Now we'll have 90 seconds for the judges to take their notes.
Okay, our second presenter is Alev, a third year in biomedical engineering. Their 3-minute thesis is titled Spatial CRISPR Screening.
Imagine a battlefield unfolding inside the human brain, where glioblastoma, or GBM, one of the deadliest forms of cancer, launches its attack on the brain. With its multiple mutations and its clever evasive tactics, it seems nearly invincible, defying our best scientific efforts and leaving patients with little hope. But what if we could crack the code of GBM's deadly agenda? What if we could pinpoint the genes, the very instructions that govern our biological function, and determine how the cancer proliferates, and develop novel therapeutic tools against them? Well, this is where my research comes in. To achieve this, we built a device that leverages the power of two technologies, CRISPR screening and dbit-seq. Think of CRISPR screening as a molecular scissors that can go inside the human body, pinpoint specific genes, and either cut them out or edit them. You could see how this would be a useful tool in determining the key orchestrators of GBM. But GBM is cunning. It exists in various environments in the brain, making it difficult to target. And this is where dbit-seq comes in, our lab's innovative engineering technology. Using this technology, we're able to look at GBM tissue sections at single-cell resolution with an additional advantage. We can preserve the spatial information. Up until this point, we've been able to look at lots of different types of cancer at single-cell resolution, but dbit-seq offers this groundbreaking advantage. Think of single-cell sequencing as a bucket of fruits with strawberries, blueberries, apples. You can reach into the bucket, pull out a fruit, differentiating it from the others, and analyze its fruity composition. This is single-cell sequencing. Now imagine that you're hosting a party, and you have a beautiful display of fruits where it really matters where each one is placed. Now, not only can you differentiate each fruit from one another, but you can also tell who its neighbors are. This is dbit-seq. It turns out that the spatial information, just like this fruit platter, is very important when we're studying complex diseases like GBM. For example, we care to understand how tumor cells interact with healthy immune cells, our body's defenders, and how those interactions change based on the environment that we're looking at. Using computational analysis tools and our new device, we're able to look at different aspects of GBM, revealing the hidden pathways leading to the disease. And we hope that we're able to narrow down the list of potential therapeutic targets to get rid of this disease once and for all. Thank you.
Thank you, Alev. Judges, you now have 90 seconds to finish scoring.
Our third presenter is Yanyu, and their presentation is The Secret Talent of the Gut Microbiome, Metabolism of Small Molecule Drugs.
When the flowers are blooming outside, you know it's the allergy season again. But do you ever wonder why one brand of allergy medication effectively clears up your friend's symptoms, but does nothing to your stuffy nose or the itchy eyes during this time of the year? The key might lie in a drug metabolism factor in our gut, known as the gut microbiome. It consists of millions of microorganisms living in our gut, and its composition differs greatly from person to person. When we take an oral medication, the drug molecules inside those pills have to pass through a whole array of these microorganisms lining our gut before entering the bloodstream and do their jobs. But the big question is, we don't really know how each type of microorganism metabolizes specific drugs, and how the corresponding structural changes to the drug molecules alter their functions inside our body. My dissertation work focuses on one type of microorganism, the gut bacteria, and my goal is to unveil how these bacteria metabolize oral drugs. To do that, I fed a commercial drug to 30 types of bacteria commonly found in the gut. And after a 24-hour waiting period, I wanted to see if and how these bacteria change this drug. First, I measured the changes in mass of the original drug molecule using an instrument called the mass spectrometer. I then determined how exactly these bacteria change the structures of this molecule through analyzing the configuration of atoms within this drug molecule. In the end, what I found was truly mind-blowing. Not only can most of these little creatures bite into the drug, but each one of them does it in a different way. Some even use the drug molecule as canvas and attach small building blocks floating around them onto the drugs. All of these structural modifications changed how well the drugs perform on their intended biological targets. No wonder why you and your friend responded differently to the allergy medication, because you might be absorbing different variants of the same drug right from the beginning. And if every individual has a unique combination of these microorganisms that can do different things to a certain drug, the resulting drug responses and potential side effects can be highly variable. My research will help doctors in the future to prescribe the best drugs just for you based on the composition of your own gut microbiome. One day, there will be no more struggles to finding the best medications for your symptoms. Thank you.
Thank you, Yanyu. And we will now take 90 seconds for the judges to take their notes.
Thank you, judges. Our fourth presenter is Jenna, I'm so sorry, who's a fifth year in microbiology. Their 3-minute thesis is titled Strike and Adipose, Eliminating Fat as a Reservoir for Q Fever.
What if I told you that I developed a technique allowing us to use light to watch an infectious disease move inside of a living organism? Well, don't believe everything I tell you. I didn't develop this technique, but I did co-opt it to study a disease or a bacteria called Coxiella burnettii, which causes a disease known as Q fever. Now, I'm sure many of you here have never heard of Q fever. It's a pretty rare disease, and most people can be treated with a course of antibiotics. So if this disease is so rare and it's treatable, what am I even doing here? Well, a small portion of people develop what we call chronic Q fever, and these people struggle with recurring bouts of Q fever even years after they cleared that initial infection. And it can lead to some pretty serious health complications and even cause death. Worse yet, we don't fully understand chronic Q fever. We don't even know from where in the body the bacteria reemerge. And that's the problem that I sought to solve with my thesis work. You can see at the top portion of my slide that the first thing I did was add a gene to my bacteria, allowing them to produce light in the same way that a firefly does. I could then infect mice with this bacteria and then use a special camera that is designed to detect the light emitted by the bacteria while inside of the mouse. And this allowed me to determine where in the mouse the bacteria were throughout the course of the infection. And using this technique, which has never before been used to study Coxiella, we found something unexpected, that Coxiella really like fat tissue. And this is interesting because some other bacteria, like tuberculosis and salmonella, have been known to hide away from the immune system inside of fat cells. And we wondered whether Coxiella was doing something similar during a chronic Q fever infection. The lower portion of my slide depicts our hypothesis that the immune system can clear a chronic or a Q fever infection, but some of these bacteria may remain hidden out inside of these fat cells, waiting for the immune system to turn its back, giving the bacteria an opportunity to reemerge and cause a new infection. Meaning that the source of chronic Q fever infections could begin inside of these fat cells. And if true, this would be a groundbreaking discovery in the treatment of chronic Q fever. But unfortunately, I have to end this talk on a bit of a cliffhanger by telling you guys how I might have found the source of chronic Q fever infections. But don't worry, the Coxiella are lighting our way to further testing our theory. We haven't yet cured chronic Q fever, but just give us a few more years. Thank you.
Thank you, Jenna. Judges, you now have 90 seconds to finish scoring.
Our fifth presenter is Arya, a fourth year in the immunology program. And Arya will be presenting on making a norovirus vaccine. All good things take time.
Norovirus, or cruise ship virus, is a nasty intestinal pathogen that causes vomiting and diarrhea. It infects up to 700 million people each year and primarily kills children under the age of 5. Despite this, we still have no norovirus vaccine. And although there are some candidates in development, the problem is they don't really work that well, meaning even if you get vaccinated, you still have a high chance of getting infected and getting sick. My thesis aims to fill this gap by understanding why candidates aren't working and also figuring out how we can make an effective vaccine that stops norovirus in its tracks. During my thesis, I've shown that the #1 protection we have against norovirus are antibodies. Antibodies are small Y-shaped proteins made by your immune system that bind directly to viruses and stop them from ever getting into your cells. But not just any antibody will do. When I tried vaccinating mice with an mRNA norovirus vaccine, I saw that they had tons of antibodies in their blood, but they were not protected from infection. On the other hand, when I tried infecting mice that had antibodies in their intestines as well, these mice were 100% immune to norovirus. This is really exciting because it seems like having antibodies in your intestine correlates with protection from norovirus. But then why were my vaccinated mice unable to make these superhero antibodies? Well, part of the answer has to do with space. An mRNA vaccine is injected into your muscle, but just because you inject something somewhere within your body does not mean that your intestines will take it seriously. This is because your gut is a really unique place where your immune system needs to be able to understand the differences between things like good and bad bacteria, food, and viruses. And it must be really, really careful not to overreact, as this can lead to really serious disease. So then it seems like the solution is easy. We just give the vaccine straight to the gut, maybe a pill, and then we'll make those intestinal antibodies. Almost, but we still have one more factor to consider: time. Norovirus replicates in the gut. When I tried infecting mice with norovirus for just 1 week, I saw that they were never able to make intestinal antibodies. But if they had an initial period of infection of 2 weeks, they made tons. This is important because it suggests that even if we give a vaccine straight to the gut, your immune system needs more than a quick first date to decide to actually mount an immune response. Taken together, my work has important implications for norovirus vaccine design. It suggests that to make an effective vaccine, we need to think in space-time, prioritizing vaccines that go directly to the gut and stick around for a while. Thank you.
Thank you, Aria. Now we'll give the judges 90 seconds to take notes.
Thank you, judges. Our sixth presenter is Mira, a fifth year in the sociology department. Their 3-minute thesis is titled The Rise of Heterosexual Refusal, Gender, Politics, and Family Change in South Korea.
Did you know that South Korea has had the lowest fertility rate worldwide every year since 2013? Those of you who watch Korean dramas here might think that romance is still in the air, but actually, data suggests that Korean adults are not only moving away from childbearing, but also from marriage and dating. The South Korean government and global media have framed this as population crisis, as you see from the newspaper headlines on the left. In line with this framework, social scientists have also asked, what can we do to make women have more children? Policies have focused on providing monetary incentives, such as tripling monthly allowances and reducing mortgage interest rates for young parents. However, even with ever-increasing government spendings, many have concluded that such policies had virtually no impact. My research takes a novel approach and challenges the existing economic explanations. Instead, I focus on the political lives of young men and women. I conducted 150 in-depth interviews with young adults aged between 20 to 39 across various backgrounds. I walked through their life histories and analyzed how political and gender values formed throughout their adulthood shaped their attitudes towards romantic relationships and family formation. My research finds that while financial factors such as job security and housing play some role, the increasing political divide and distrust between men and women shape their decisions around heterosexual relationships. First of all, women are becoming more liberal and feminist, whereas men are turning from feminism and becoming enormously conservative. And I find that this political divide has an important influence on young women to opt out of heterosexual relationships. Secondly, some women are refusing heterosexual relationships as a direct response to how the government and researchers place responsibility on women for low fertility while omitting their voices. One of the participants said, if I give birth to a child, that means I'm helping to increase the fertility rate for the country, right? I'll never do that, especially because the South Korean government thinks of women having kids as a way to boost economic development. If fertility continues to decline, Korea's population will shrink by 30% in less than 50 years. And this problem has begun to haunt other countries as well. While my research emphasizes the gap between the government's strategy and the social reality of young adults, I further highlight that policymakers and researchers must pay attention to the gendered political divide as a way to understand changing desires and attitudes towards heterosexual relationships. Thank you.
Thank you, Mira. Judges, you now have 90 seconds to finish scoring.
Now we are ready for our seventh presenter, Li Ha, who will present on understanding the language of neurons.
Waves are ubiquitous in nature. Light is a wave. Sound is also a wave. Actually, sound is a traveling wave. It travels from my mouth to your ears. Traveling waves contain information. The information contained in the sound wave is called language. Understanding human language can help us understand how the human society works. Similarly, neurons talk to each other with their own language. And understanding the language of the neurons can help us understand how the brain works. Once we understand how the brain works, we can understand cognition and use this knowledge to cure brain diseases like Alzheimer's. My thesis research seeks to understand the language of the neurons by studying the traveling waves. In our lab, we focus on the visual cortex of macaques. We choose macaques because their visual cortex is similar to ours. The visual cortex of macaques has layered structures. We study the traveling waves along the depths of the layers because we want to know how neurons in different layers talk to each other. So far, people have only studied traveling waves along the surface. We collect electrical signal data by putting a needle into the visual cortex. First, we want to know whether the traveling waves exist. With human language, you know I am speaking because speech follows distinct patterns. Applying exactly this simple idea, we look for traveling wave patterns that are impossible to exist in random noise. We indeed find them, so we conclude that the traveling waves exist. Next, we want to know what information is contained in the traveling waves. Human voice has low pitches and high pitches, reflecting different emotions. We analyze the pitches of the traveling waves and find that there are low-pitch traveling waves when the macaque says nothing. On the other hand, there are high-pitch traveling waves when the macaque says a flash. Therefore, we conclude that the traveling waves contain information about vision. In summary, my research shows that neurons in different layers of the visual cortex talk to each other using the traveling waves, which contain information about vision. My research is just one small step towards understanding how the brain works. But with the collaborative effort of the entire science community, we can one day understand cognition and cure brain diseases like Alzheimer's. Thank you.
Thank you, Li Hao. Now we will have 90 seconds for the judges to make their judgment.
Our final presenter is Leonardo, a 6th year in economics. Their 3-minute thesis is titled, "The Cost of Dropping Out of High School, Why Every Diploma Counts."
Imagine waking up in one day and deciding you're not going to school anymore. This is Lucas' story, a Brazilian guy who left school to work on a farm. His story is unique though. In Brazil, 40% of students never finish high school. Even in the US, this number is 13%. The cost of dropping out is much more than just missing graduation days. Those individuals are more likely to live in poverty, committing crimes, and suffer health problems. But what if we could change Lucas' story? What if a simple intervention could change the future of millions like him? So my research investigates the long-term impacts of a government cash transfer program to high school students in which they receive a financial reward for each year completed in school. This is not just an idea. 5 years ago, Brazil, Colombia, and Mexico launched some pilot programs in small communities, offering students up to $200 a year to encourage school attendance. And those programs have reduced dropout rates by 40%. But still, what happens after graduation in terms of wage, unemployment rate? And if we want to expand these programs nationwide, does it lead to higher economic growth? Unfortunately, we don't know. And since those programs are new and implemented in small communities, we don't have enough data. To answer those questions, we propose a different method. We build an economic model that simulates the Brazilian economy and all students' decisions over their lifetime. By doing that, we can answer all those questions. Our results: a large-scale program increases wages in the economy by 5% and decreases unemployment rate by 15%. But most importantly, by these gains, it leads to a higher Brazilian GDP of 3%. And surprisingly, when we simulate the same program, but targeting college students instead high school students, the results are much lower. And this is important because Brazil spends 4 times more funding to college students than to high school students. So our results suggest that targeting high school students would lead to higher social benefits, right? But this isn't enough. Our job isn't finished yet. We need to transform these findings into real changes. And we're doing that. How? Well, besides being a PhD student in economics, I'm also a congressman in Brazil, and I have transformed this policy into a legislation. So this is me working on in this bill. So I hope to come back here one day to implement this program, which was born here, so that we can have no more stories like Lucas. Thank you.
Thank you, Leonardo. Judges, you have 90 seconds to finish scoring.
Okay, that concludes all of our presentations. So let's give our presenters a round of applause.
Thank you. The judges will now leave the room to deliberate for 20 minutes while you, the audience, votes for your favorite presentations.
We're welcoming Lynn up to the stage to announce our 2 People's Choice winners and our 3 3-minute thesis winners.
Okay, hi everyone. Thanks so much for being here. Thank you again to our moderators here. Did they do a good job or what? Okay, I'm going to say thank you to the judges next. You guys, you made this so hard to figure out. So I want to say congratulations to all of you for beautiful presentations and very interesting research projects. I hope this exercise was helpful to you in transmitting the excitement you so clearly have about your work. So thank you. Great hand for all of them. So I was saying on the way over to my colleague judges that I've sat on several award committees, and the good news is you get down to the final decisions and really you can't make any bad ones. The bad news is it's really hard to actually make decisions. So, but we did, and you did, so I can now tell you the results. Okay, People's Choice Awards first, so these were voted on by all of you, and there are 2. And one is Alev Baysoy. Who talked to us about spatial CRISPR screening. Congratulations. The second is Leonardo Di Sequeiro Lima, who talked to us about how to keep kids in high school. So thank you. Okay, and now on to the, oh, just them, right? Oh. Good. Okay, don't go away. Okay, and then in 3rd place is Yanyu Zhao. Who told us about the secret talent of the gut microbiome. Oh, come over here. How about that? Okay. So don't go, just stay right there, because we'll get everybody at the end. And then in 2nd choice, Leonardo Di Sequeiro Lima. And these are look like checks because they're real money that they'll get, so just not these. Okay. Okay, and then I wish I had a drum roll. So drum roll for, oh, awesome. For our first place winner this year of the 2024 3-minute thesis contest, Arya Akin. Microphone for just a minute.
Oh man, I don't know what to say. I didn't expect to be here. Thank you. Thank you so much to everyone who came both to support the competition. I think it's really important for us to learn how to communicate our science and other topics, obviously, as well, but to communicate ourselves to people, because that's the only way to really make your research go forward, is to make sure that people know about it. And special thank you to everyone that came out to support me. I really appreciate all of you and your friendly faces in the audience. Thank you.
Congratulations. That's enough pictures. Okay, congratulations to all of you. Congratulations to all of you for participating in this year's contest. And all of you in the audience, do this next year. We need more people to participate in the 3-minute thesis. So I'm going to hand this back to our moderators, and thank you again all for being here.
Congratulations again to all of our winners and to our finalists for their amazing presentations this evening. Please, before you guys head out, we're just going to do a few thank yous. So we just need a few more rounds of applause for the various offices that made this event possible. So please join me in thanking the Office of Career Strategy and the Graduate Writing Lab for supporting our participants with coaching on public speaking and slide design. Also join me in thanking the technical support from the Yale Broadcasting and Classroom Technology and Events for this hybrid event. They're in the back. Please give them a round of applause. Yes. Also join me in thanking our esteemed panel of judges and all 12 of our preliminary round judges. We also thank GSAS, particularly Dean Cooley, and our alumni, especially those on the GSAA board, for supporting 3-minute thesis this year and each year after this. Let's give them a round of applause as well.
And of course, we'd like to give a special shout out to Suzanne's office, who organized this event, the GSAS Office of Professional Development. And I'd like to encourage all graduate students to take advantage of Suzanne. She's here to advise you on your professional development throughout your PhD. And of course, again, we'd like to encourage everyone to attend Where Do I Go from Yale, which is the event that's starting tomorrow. And thank you. Oh, Suzanne.
And just to prolong these a little bit longer, I want to thank Roxanne, Joe, Ming, and Catherine, my 4 fellows this year. I've had the great pleasure of working with them all year, and we've come to this important public-facing event, and they've helped me pull it off flawlessly. So, thank you.